Good morning. In this video, we will be discussing about the pathway of gluconeogenesis. Normally, the diet we take in can maintain the blood glucose level for some 4 to 6 hours. That's the reason exactly after 4 to 6 hours after breakfast, you are taking lunch. And again after 6 hours from lunch, you are taking your dinner. Suppose if we are missing any of these meals, what will happen means the body will start producing the glucose either by glycogenolysis or by gluconeogenesis. In the initial hours, the source of the glucose will be by glycogen breakdown. But the glycogen content of the liver can manage the blood glucose level only for around 10 to 12 hours. So beyond that, if you are fasting, our gluconeogenesis will take the main seat. It will be becoming the major source for maintaining the blood glucose. So what is this gluconeogenesis? How it takes place, where it takes place, all these things we will be discussing in this video. Gluconeogenesis, the name itself says it is nothing but the synthesis of glucose. Genesis means synthesis, glucose is glucose. Okay, so the synthesis of glucose from non-carbohydrate sources is called as gluconeogenesis. That means here we are excluding the glycogen, because from glycogen also glucose can be synthesized, but that comes under glycogenolysis. Okay, so in order to remove that, we have kept the specific term from non-carbohydrate sources. The major non-carbohydrate sources which can be used for glucose synthesis are lactate, glucogenic amino acids, odd chain fatty acids, and glycerol. So these are the major 4 non-carbohydrate sources from which glucose can be synthesized, and this process is called as gluconeogenesis. This process, it actually takes place exclusively mainly in the liver. Why only in the liver means one of the enzymes of this pathway, glucose 6-phosphatase, one of the key enzyme in gluconeogenesis pathway is present only in liver. That is the reason gluconeogenesis takes place mainly in the liver. But in addition to the liver, a minor portion can also happen within the renal cortex and also in the intestine. Okay, but mainly gluconeogenesis, the major source for glucose or gluconeogenesis is only liver. Okay, so now within this liver, how this gluconeogenesis takes place, how the pathway takes place, we will discuss in detail. The pathway of gluconeogenesis can be broadly classified into 2 phases. In the first phase, what we have to do is, I mentioned about the various sources, right? So these sources or these substrates will be converted to any one of the glycolytic intermediate. That is the first phase. All these sources will be converted to any one of the glycolytic intermediate. Then once it has entered into the glycolysis as a glycolytic intermediate, the second phase is just the reversal of glycolysis to produce glucose. Okay, so this is how gluconeogenesis takes place. In the first phase, the substrates, the non-carbohydrate substrates, okay, sources will be converted to, somehow it will be converted to glycolytic intermediate, and from there the glycolysis will be reversed and it will be converted to glucose. Okay, so now we will see about the first phase, how the various non-carbohydrate sources are converted to glycolytic intermediates. First we'll start with lactate, lactic acid. So normally this lactic acid is produced as a result of anaerobic glycolysis. Okay, so under anaerobic conditions, glucose will be initially converted to pyruvate. That pyruvate will be converted to lactate by lactate dehydrogenase enzyme. Okay, so this reaction actually is a reversible reaction. So the same enzyme lactate dehydrogenase will convert the lactate back to pyruvate during starvation condition. Okay, so this is how lactate is converted to pyruvate, which is one of the glycolytic intermediate. The major source for lactate, so during starvation for gluconeogenesis, is from the liver, from the muscle glycogen. Okay, so muscle, what will happen in like liver, in muscle also glycogen is stored. Okay, liver glycogen can directly participate in the glucose production during starvation time, but muscle glycogen cannot be converted because the enzyme glucose 6-phosphatase is absent in the muscle. So what will happen means within the muscle, the glycogen will be broken down, it will be converted to pyruvate, and that pyruvate will be converted to lactate. Okay, so glycogen. Okay, so here please don't, it is not glucose, it is actually glycogen. Okay, so glycogen, when it breaks down, it forms glucose 1-phosphate 6-phosphate. From that pyruvate will be formed, and that from that pyruvate lactate will be formed. So now that lactate formed in the muscle will come out of the muscle and enter into the liver, where they will be converted to pyruvate, and that pyruvate in turn will be utilized for gluconeogenesis. Okay, and this glucose thus formed from the liver can be utilized by the other tissues including the muscle also, where there can be again converted into lactic acid. Okay, so this interconversion of lactic acid to glucose and glucose to lactic acid is called as lactic acid cycle, or otherwise called as CORI'S cycle. So this is in brief about how the lactic acid, the intermediate, how lactic acid is converted to glycolytic intermediate pyruvate. Next, coming to the second source. The second source is glucogenic amino acids. So totally we have around 20 amino acids. Among them, except leucine, all the other amino acids are glucogenic amino acids. So in that, some are partly glucogenic, whereas some are purely glucogenic. Okay, for example, lysine, isoleucine, and all the aromatic amino acids are partly glucogenic, whereas the rest all are purely glucogenic in nature. So all those amino acids can be used for gluconeogenesis. But among all those amino acids, the most commonly used amino acid for gluconeogenesis is alanine. Okay, this alanine is the most commonly used amino acid for gluconeogenesis. So how this is converted to glycolytic intermediate means by a process called as transamination reaction. Okay, so in this transamination reaction, what will happen? From alanine, the amino group will be transferred to alpha ketoglutarate, so that what will happen? The alanine, the amino acid, after losing its amino group, it will be converted to its keto acid pyruvate. In the same reaction, alpha ketoglutarate will be converted to glutamate. Okay, so what is happening? Alanine, by transamination reaction with the help of the enzyme alanine transaminase, it will be converted to pyruvate, which is actually one of the glycolytic intermediate. Okay, so this is how alanine is converted to glycolytic intermediate. Okay, so like lactate, this alanine also mainly comes from the muscle protein breakdown. So during starvation, what will happen? Muscle proteins will start breaking up, and they will be releasing the amino acids, and that amino acid, especially the alanine, it will undergo transamination to pyruvate, and that pyruvate will be reaching the liver, where it will be converted to, where it will be utilized for gluconeogenesis. Okay, so this is how alanine is converted. So coming to the other amino acids, so how they will enter into gluconeogenesis means, so among the other amino acids like alanine, few other amino acids like cysteine, glycine, serine, threonine, and tryptophan, all of them will be forming pyruvate by a simple transamination, not by simple transamination, by one or other way they will be forming pyruvate, and thus it enters into the glycolysis. Whereas the other amino acids, they will be forming the TCA cycle intermediates during their catabolism. Okay, so when it forms the TCA cycle intermediates, they will be ultimately converted to oxaloacetate. Okay, that oxaloacetate in turn, it will be converted to phosphoenol pyruvate by the enzyme called as phosphoenol pyruvate carboxykinase. Okay, so once it forms phosphoenol pyruvate, it is nothing but the glycolytic intermediate. Okay, so some amino acids, they will directly enter as pyruvate, and some amino acids, they enter the TCA cycle, they form oxaloacetate, and from oxaloacetate it will be forming the phosphoenol pyruvate. So this is how the glucogenic amino acids enter into the glycolysis. Okay, so next, the third source, odd chain fatty acids. So odd chain, even chain fatty acids, during their catabolism or oxidation, they will be producing 2-carbon acetyl-CoA molecules. So this acetyl-CoA molecule cannot be converted back to pyruvate or any other TCA cycle any other glycolytic intermediate, because the enzyme, the pyruvate dehydrogenase complex enzyme, is strictly reversible. Okay, and the other thing is, by the time if it goes in TCA cycle, it will be completely oxidized there before it reaches the oxaloacetate. Okay, so because of these two reasons, the odd even chain fatty acids cannot be utilized for gluconeogenesis. But the odd chain fatty acids, which will be having odd number of carbon atoms, during their oxidation, they will be finally forming a 3-carbon propionyl CoA. Okay, so that 3-carbon propionyl CoA can be converted to succinyl CoA, which is again a TCA cycle intermediate. Okay, so once it forms a TCA cycle intermediate, like through TCA cycle, it will be forming oxaloacetate, and that oxaloacetate in turn will be converted to phosphoenol pyruvate. Okay, so this is how odd chain fatty acids enter into glycolysis. It enters as a glycolytic intermediate. Okay, so it enters the at phosphoenol pyruvate point. Okay, so that is how it enters. So first odd chain fatty acids, they will be forming propionyl CoA. Propionyl CoA, with the help of B12 dependent enzyme, it will be forming succinyl CoA. That succinyl CoA in turn will be forming oxaloacetate, and that oxaloacetate will be entering the glycolysis as phosphoenol pyruvate. Okay, so that's it about the third source. Now coming to our last source, glycerol. Okay, so how this is converted means very simple. Actually, the other three things, lactate, it entered as pyruvate. Alanine, it entered as again as pyruvate. The other amino acids and odd chain fatty acids, they entered at phosphoenol pyruvate. They all entered almost at the terminal reactions of glycolysis. But glycerol, they enter in the almost the initial reactions of the glycolysis. So this glycerol, how it enters means they enter as dihydroxyacetone phosphate. They enter the glycolysis as dihydroxyacetone phosphate. So how glycerol is converted to dihydroxyacetone phosphate means initially this glycerol will be phosphorylated by glycerol kinase enzyme to form glycerol 3 phosphate. Next, in the subsequent step, this glycerol 3 phosphate will be oxidized and it will be converted to dihydroxyacetone phosphate by the enzyme glycerol 3 phosphate dehydrogenase. Okay, so this is how glycerol is converted to glycolytic intermediate dihydroxyacetone phosphate. Okay, so now with this, phase 1 is over. We have converted all the non-carbohydrate sources. That means lactate, glucogenic amino acids, odd chain fatty acids, and glycerol. So all these things we are converted into a glycolytic intermediate. Okay, so now what is the next phase? It is just the reversal of the glycolysis. So among the 10 reactions of glycolysis, almost 7 reactions are reversible reactions, so they can be easily reversed. Okay, only 3 reactions are irreversible. Which are the irreversible reactions means? One is hexokinase step, the reaction catalyzed by hexokinase, the first step. The third step, the reaction catalyzed by phosphofructokinase, and the last step, the reaction catalyzed by pyruvate kinase. Okay, so these are the 3 kinase reactions in the glycolysis which are irreversible. Okay, so if you produce an alternate enzyme for these 3 enzymes, you can easily reverse the glycolysis. So now we will see what is the alternate enzyme for these 3 reactions. So first we start with hexokinase. So in glycolysis, what is the action of this hexokinase means? It will phosphorylate glucose and it will be converting it into glucose 6 phosphate. This is the normal action of hexokinase. Okay, so now in gluconeogenesis, this reaction has to be reversed. So in the reverse, what is happening? Glucose 6 phosphate, the phosphate from glucose 6 phosphate will be chopped and removed so that glucose will be formed. So this reaction in which a phosphate molecule is removed from glucose 6 phosphate is called as glucose 6 phosphatase. Okay, so that is the alternate enzyme of hexokinase in the gluconeogenesis pathway. So now coming to our one special thing about this glucose 6 phosphatase, in the initial slide only I told this glucose 6 phosphatase is present mainly in liver. That is the reason gluconeogenesis takes place mainly in the liver. Okay, so this enzyme is present only in the liver endoplasmic reticulum. That's why gluconeogenesis takes place only in the liver. Okay, so first reaction we have reversed. Next, the second irreversible reaction is phosphofructokinase. What is the action of this enzyme means? It will convert fructose 6 phosphate. It will add a phosphate group. Any kinase, it will result in phosphorylation reaction. Okay, so it will add a phosphate to fructose 6 phosphate and converts it into fructose 1,6 bisphosphate. This is what the reaction catalyzed by phosphofructokinase in glycolytic pathway. Now the same reaction will be reversed. Okay, so in the reverse, what is happening? Fructose 1,6 bisphosphate will be broken down. One phosphate will be removed. Okay, so here the enzyme for such a reaction is called as fructose 1,6 bisphosphatase. So this enzyme will help in the reversal of the step catalyzed by phosphofructokinase. Next, coming to the third step, the last irreversible step, which is catalyzed by pyruvate kinase. Okay, so what is the action of this pyruvate kinase means? It will convert phosphoenol pyruvate to pyruvate. That is the action of pyruvate kinase. Okay, so now what we are going to do is we have to convert pyruvate back to phosphoenol pyruvate. Unlike the other 2 reactions, this is not as simple. Okay, this is not a simple one-step reversal. Okay, so here it will involve multiple steps. Okay, so what are those multiple steps? I will explain you now. So initially, what we have to do is actually the reaction. First we have to convert pyruvate into oxaloacetate. We cannot directly convert pyruvate to phosphoenol pyruvate. Instead, first pyruvate should be converted to oxaloacetate, and then the oxaloacetate will be converted to phosphoenol pyruvate. Okay, so this is how the reaction, the reversal happens. Okay, but again there is one small complication in it. What is that complication? Means the initial reaction pyruvate to oxaloacetate, the enzyme for this reaction is present in the mitochondria. But whereas the next reaction where the oxaloacetate will be converted to phosphoenol pyruvate, that happens within the cytoplasm. Okay, so what we should do is initially the pyruvate which is formed in the cytoplasm should be transported to the mitochondria, and then within the mitochondria pyruvate will be converted to oxaloacetate, and then the oxaloacetate formed in the mitochondria is be transported to the cytoplasm, and then within the cytoplasm the oxaloacetate will be converted to phosphoenol pyruvate. Okay, so this is the a brief thing how the pyruvate kinase step is reversed. Okay, so now we will see in detail about all these steps. So what is the first step? The pyruvate should enter into the mitochondria. Okay, so how this will happen means in the mitochondria we have a specific transporter called as pyruvate transporter, which will allow the pyruvate to enter from cytoplasm into the mitochondria. So once it enters the mitochondria, it will be converted to oxaloacetate. So how this is happening means actually pyruvate is a 3-carbon molecule, whereas oxaloacetate is a 4-carbon molecule. Okay, so how to convert a 3-carbon to a 4-carbon? Just by adding 1 carbon to it. Okay, by a carboxylation reaction which is catalyzed by the enzyme pyruvate carboxylase, for which biotin and ATP acts as a coenzyme. Okay, by a simple carboxylation reaction we can convert pyruvate into oxaloacetate. Okay, so now this oxaloacetate is within the mitochondria, but the enzyme which can convert it into phosphoenol pyruvate is present only in the cytoplasm. So next we will see how this has to go out to the cytoplasm. Okay, unfortunately, like pyruvate transporter, we don't have any specific transporter for oxaloacetate which can easily transport the oxaloacetate from mitochondria to cytoplasm. Okay, so we need a shuttle pathway. Okay, we need a bypass pathway which is nothing but malate aspartate shuttle. So the oxaloacetate formed in the mitochondria should get transformed, should get disguised either as malate or aspartate, and sorry, here it will form in the mitochondria, so it should disguise itself either as malate or aspartate, and then it should go out of the mitochondrial membrane into the cytoplasm. Okay, so whether to go out as malate or to aspartate is decided by the source or the substrate from which the gluconeogenesis process is started. Okay, so normally what will happen means normally for most of the substrates oxaloacetate prefer to go as malate. Okay, so oxaloacetate will form malate, and that malate through malate transporter will go out, and that malate in turn in the cytoplasm will be converted back to oxaloacetate. Okay, so why this pathway is preferred, why malate pathway is preferred means, so during this process along with oxaloacetate one more NADH is also transported into the cytoplasm. Okay, so this NADH is very much needed for reversal of the glycerol 3-phosphate dehydrogenase step in gluconeogenesis. Okay, that's the reason malate shuttle is always preferred by oxaloacetate. Okay, but if the substrate, the source for the gluconeogenesis is lactate means, so during the conversion of lactate to pyruvate itself, one NADH molecule will be formed. So we don't need one more NADH molecule from the shuttle pathway. So under such circumstances, it will try to come out as a aspartic acid. Okay, so if the starting point is lactate, the glucose is synthesized from lactate, the oxaloacetate will be using the aspartic acid pathway, whereas for the other substrates it will be using the malate pathway. Okay, so anyhow, though in short, it is using the oxaloacetate is using malate aspartate shuttle to come out of the mitochondria. So once it comes out to the cytoplasm, by the same enzyme phosphoenol pyruvate carboxykinase, it will be converted to phosphoenol pyruvate. Okay, so this is how the pyruvate kinase step is reversed. Okay, so it is a quite complicated process. It actually takes place in 2 steps, but in between few shuttling processes are also involved. Okay, so what are the 2 enzymes which are needed for replacing the action of pyruvate kinase in gluconeogenesis means, one is pyruvate carboxylase, which converts pyruvate to oxaloacetate, and the other one is phosphoenol pyruvate carboxykinase, which converts oxaloacetate to phosphoenol pyruvate. Okay, so once all these steps are reversed, so with the help of these enzymes, that means glucose 6-phosphatase, fructose 1,6 bisphosphatase, pyruvate carboxylase, and phosphoenol carboxykinase. Okay, with the help of these enzymes and other reversible enzymes of glycolysis, the pathway of glycolysis can be easily reversed and it can be utilized for glucose synthesis, gluconeogenesis. Okay, so this is how the pathway of gluconeogenesis takes place. Okay, so normally the pathway gluconeogenesis is nothing but the synthesis of glucose from non-carbohydrate substrates like lactate, glucogenic amino acids, or chain fatty acids, and glycerol. Okay, so all these sources, first they will be converted to glycolytic intermediate. Lactate will be forming pyruvate, glucogenic amino acids, few will be forming pyruvate and few will be forming phosphoenol pyruvate, or chain fatty acids will be forming phosphoenol pyruvate, and glycerol will be forming dihydroxyacetone phosphate. Okay, so once all of them enters into glycolysis, just by the reversal of glycolysis, by the alternate enzymes for the reversible irreversible enzymes, easily the pathway can be reversed and we can easily get the glucose formed in the liver. Okay, so that's it about the pathway of gluconeogenesis. So the only thing, one more thing which you should know about this pathway is regulation. Okay, so how the gluconeogenesis is regulated means it is just a reciprocal regulation of glycolysis. Okay, so both the regulation of glycolysis and gluconeogenesis together we will discuss in the next video. Thank you.